Hello everyone, this is Savannah. We're doing genetics worksheet number 2. Like last time, we have our lecture review section and then we'll have some practice questions at the end. So let's start with question 1. So the first question is, what are some differences between prokaryotes and eukaryotes? In a broad sense, the biggest difference between eukaryotes and prokaryotes is that eukaryotes are far more complex. What do we mean by complex? Well, these have organelles. And as we saw later, the DNA replication and the production of proteins is going to be far more complex than prokaryotes. And the other big difference is that prokaryotes don't have a nucleus. They have something called a nucleoid, which is just a region where the genetic material is at. So it's not like in a eukaryote, let's say, here's my cell, eukaryote would have a nucleus and it'd be contained, while in a prokaryote, it would be just a general region of where the DNA is. That's a big one, just know nucleoid versus nucleus. And of course, eukaryotes are far more complex. Okay, so the next question is circle the following with the correct associations. So are bacteria prokaryotes or eukaryotes? If you thought eukaryotes, you would be correct. Protists, are they eukaryotes or prokaryotes? If you guessed eukaryote, you'd be correct. Plants, are they prokaryotes or eukaryotes? Eukaryotes, that one's kind of an easy one. Fungi, are they prokaryotes or eukaryotes? These are eukaryotes. Archaea, are they prokaryotes or eukaryotes? Prokaryotes. Remember, archaea are going to be our extremophiles. So ones that live in like acid or under super pressure or high temperatures, stuff like that. So archaea are prokaryotes. Number 3, what are the genotypic and phenotypic expressions for homozygous recessive, homozygous dominant, and heterozygous? So what they mean by genotype is going to be our codes, which are typically going to be represented as our alleles. So homozygous recessive is going to be little a. So this is going to be genotypically what it looks like. And then phenotypically, we're going to express the recessive trait. I'm spelling this so wrong. Recessive. Okay. I probably spelled that wrong. I'm terrible at spelling, but we'll just, you know what I mean. Okay, now homozygous dominant. This is going to be genotypically big A, and this is going to be the dominant trait. And then lastly, we have heterozygous, which is genotypically big A, and most of the time it's going to be expressing the dominant gene. We'll see later on there are exceptions, but in all of exam 1 questions, it'll likely be that there are only talking about heterozygous big A, or big A, and it's going to be the dominant trait. We'll see exceptions in later chapters, but for now, let's just say phenotypically it's dominant. Okay, number 4. What is cytogenetics? Cytogenetics is a field in genetics that explores microscopic examination of the chromosomes. So just say microscopic examinations of chromosomes. This is just a fun little definition that might be a testable question. If anything, you just need to know the definition of it, right? So when we're talking about people studying chromosomes, or we're even just like how we learned what chromosomes, what they do, what's functioning with them, it would be from cytogenetics, the microscopic examination of chromosomes. Okay, number 5. This one's pretty important. This is a very important concept, and it'll build up to more higher level questions later on. And we need to set this foundation of what is a gene loci. So loci, I want you to think location. So this is the actual physical location of the genes found on the DNA and chromosome. We'll just say actual physical location of genes, right? Gene loci, right? This is just a definition. Memorize it. Super important. It'll be super important later on. Might as well learn it now. All right, number 6. This is kind of a big, broad question. So what is the cell cycle? So the cell cycle, right, is we have this thing called interphase. Right, interphase is going to be made up of G1, which is our growth. Then we're going to have S phase, which is DNA replication. Then we're going to have G2, which is more growth. And then we'll have mitosis. And then sometimes it gets lumped in with cytokinesis. Cytokinesis, right? That sometimes get lumped in with mitosis. Interphase is all of these guys. And then we have mitosis and cytokinesis. Sorry I didn't make that terribly clear. And then again, let's just go over mitosis real quick, right? We have prophase. In the book, they talk about prometaphase. Depending on what textbook, sometimes they get lumped together. It's just prophase or, yeah. And then metaphase, which is when they line up on the plate. And then we have anaphase, which is when they get pulled apart. And then we have telophase, which is when they get more, not completely separated, but they start forming this. And then the actual cleavage of it is going to be cytokinesis, which is going to be a cleavage furrow for eukaryotic cells. All right, just an overview of that. Yeah, there will be questions that will have you refer to some information dealing with the cell cycles. Just be aware of the broad concepts of it. And we'll get into more of the focal points of those phases. All right, question number 7. What is the importance of kinetochores? So these are kind of important. I think this is a highly testable topic. Kinetochores are the fibers that actually bind to the chromosome at the centromere. And it's super important because it allows the chromosomes to be moved. So say we have a chromosome here, right? So our centromere is going to be the center part right here. And our kinetochores, so I'm just going to add a different color. So we have these organelles called centrioles. And we have, this is where our microtubule organization kind of emanates from, our center, if you will. And these kinetochores are going to attach to the centromere. And this allows for us to move them. So kinetochores, just to put it in words, we'll say fibers that actually bind to the chromosome at the centromere. Right, and there are other factors too that deal with the movement of chromosomes. We'll see that later in question 9, but kinetochores are going to be your big ones. Again, main part is they attach to the centromere. All right, question 8. What is the difference between sister chromatids and homologous chromosomes? So sister chromatids are identical copies of the chromosome, while homologous are similar copies of the chromosome, one which you get from your mom and one which you get from your dad. So a lot of the times it'll show sister chromatids like that, and then homologous chromosomes will be a pair. Right, and this will be pretty clear when we're looking at meiosis, we'll see sister chromatids lining up in meiosis 1, and then we'll have, 2, and then homologous chromosomes will be meiosis 1. Oops, spelled that wrong. And then these homologous chromosomes are where we're actually going to see crossing over, and the synapsis, which is synapsis is the gluing of the chromosomes together, and then crossing over is the actual exchange of material between the chromosomes. Okay, so question 9. What is the MTOC, and what are the 3 types of microtubules? So the MTOC stands for the Microtubule Organization Center. So, Microtubule Organization Center. All right, and so as the name suggests, it organizes microtubules. And then there are 3 types that your book mentions for the microtubules. The first one is called astral microtubules. So these are going to help position the spindle apparatus. Spindle apparatus kind of encompasses the centrioles and the kinetochores that attach to the centromeres. That's the broad view, we call it the spindle apparatus. And then our second one is going to be our polar microtubules. So our polar microtubules help push the poles away from each other. Right, so whenever we're going through anaphase, we need something that will, or even before anaphase, we need these centrioles, right? This is before our polar, I guess. So we have these centrioles, which the spindle apparatus grows from. We need these to be on opposite sides. So these polar guys will pull this one that way, this one this way, so that at the end we have an apparatus on this side and an apparatus on this side. So we can have the metaphase plate, and we can have anaphase, which is going to push these even further so that the sister chromatids get separated. Okay, so then the last one we already mentioned, this is going to be your kinetochores. Right, and the main thing that you know is this is the microtubule that actually attaches to the centromere. Right? Okay, question 10. What happens during zygotine and pacotine? I don't know if I'm saying the P one correctly, so sorry if I mispronounced that. But so these are referring to a group of phases that happen in prophase. So prophase has a bunch of just like subset levels or subset steps, if you will, within prophase. So the first one that we're going to talk about, there's a whole bunch of them. You only need to know zygotine and pacotine. Sorry about that. So zygotine is going to be where the synaptonemal complex forms and synapsis occurs. /synapsis. Right, so what do I mean by synapsis? So you have chromosome from mom, your chromosome from dad, and there's this binding that can happen between the chromosomes. So this is like where the chromosomes gluing together. So as a result, you would have your mom chromosome, your dad chromosome, basically stacked on top of each other. And this connection is going to be called our synaptocomplex. This whole step, this process we refer to as synapsis. Okay, so then Pachytene. Pachytene is going to be when homologs are now aligned. This forms the bivalence, and the bivalents now cross over and exchange genetic material. So bivalence, remember, that's just going to be our mom and dad chromosome stacked on top of each other. This is our bivalent, also called our tetrad. And pachytene is going to be crossing over. This is highly, highly testable. I'd put a big star next to it. This is just simple memorization. Zygotene, synapsis, pachytene is going to be crossing over. Okay, so number 11. What is the difference between crossing over, synapsis, and chiasma? So we already said crossing over is the actual exchange of, the physical exchange of chromosome genetic material. Right, and then synapsis is going to be the homolog chromosomes recognize each other and begin to align themselves along the lengths of each other. So we'll just say, I like to think of it as they glue each other together. Right, so these chromosomes are going to glue each other together first with the synaptic complex, and then they're going to cross over and exchange genetic material. So synapsis happens first, then crossing over. And chiasma is the actual physical site where chromosomes cross over. Physical site. Oh, I think that's what that is. Where chromosomes. Oh, I cannot spell or speak tonight, apparently. Physical site where chromosomes exchange material. Sorry about that. Okay, question 12. What are the end products for mitosis and meiosis? This is a super important one. I put a star next to it. This is a highly testable topic. Right? We should know this. This should be reviewed, but in case you don't, mitosis, we're going from, and let's use humans as an example. We're going from 46 chromosomes, which is 2N, and we're going to two cells. So it kind of looks confusing. We're going to have one cell that's 46, 2N, and we're going to have another cell, which is the same. So we're going from one diploid cell to two diploid cell. And then meiosis, we're going from 46 to N, that's our starting thing. And then we're going to 23 N. And then these are going to go through another phase. I didn't give myself enough room. Oh. So I'll just highlight the end products because it's a little confusing. So for the mitosis, we're going to have two diploid cells. And then for mitosis, or meiosis, we're going to have 4 unique haploid cells. So again, I'll just draw this out a little neater. Sorry if this is confusing. Mitosis, 2 identical daughter cells. Meiosis, it's going to be 4 different daughter cells. This is going to be highly testable. you'll probably be tested comparing meiosis and mitosis. There's an incredible table in the book. I would go over that. Just look over it. And then if you have any quiz questions that talk about meiosis or mitosis or assignment questions, I would go over those because meiosis and mitosis will be highly testable on exam 1. Okay, so now we'll go ahead and get into our practice questions. Okay, question number 1. Mitochondria and chloroplasts have their own DNA. True or false? Correct answer is true, right? Mitochondria and chloroplasts have their own DNA. They even have their own membranes. This goes back to the idea that mitochondria and chloroplasts were engulfed into the cell via endosymbiosis. So yeah, they have their own DNA. Number 2, the X and Y chromosome are homologous chromosomes. True or false? The correct answer is false, right? Homologous chromosomes are going to be very equal in length, right? For the sex chromosomes, the X chromosome is really big, and our Y chromosome is very small. And in fact, it's getting even smaller. It's shrinking. So yeah, X and Y are not homologous chromosomes, and later we'll see that there's a compensation dosage that happens with females because females will have too many chromosomes, too many genetic material as opposed to males. So yeah, X and Y chromosomes are not homologous. Question 3. Binary fission produces the correct answer is B. So right, there's binary fission is the way that bacteria divide. So bacteria goes like that, and then it just kind of splits in half, and you have 2 exactly the same bacterial offspring. So binary fission, identical bacterial offspring. Meiosis 1. Just finish the sentence. The correct answer is E, right? Meiosis 1 goes from diploid to haploid, right? We saw that in this disgusting diagram. We went from diploid to haploid. Yes, meiosis 1. And then C is also correct. It is called a reduction phase. Something you do need to know, highly, highly testable. Meiosis 1 is our reduction phase. And then meiosis 2 is our equational phase. Right? Highly, highly testable. Okay, number 5. Random independent assortment is. I'm going to put a big star next to this because this is also highly, highly testable. The correct statement is, let me get my highlighter. Number C, or letter C, the random alignment of bivalence during meiosis 1. So this is highly testable. This independent assortment and independent segregation are going to be super tested, highly testable. This is just an easy point on the exam if you just know the definition of it, right? So what it means by random alignment of bivalence, if you can recall, we said bivalence were this collection of synaptal or synapsis chromosomes, and we're just going to randomly align them along the metaphase plate. So that red line is the metaphase plate. These are chromosomes. They're just going to randomly align, and that creates genetic variation because this one goes over there, this one goes over there, but if they were switched, then it would be different. So yeah, know this definition, highly, highly testable. Question 6. What structure has to form in order to actually exchange genetic material during prophase 1? Sorry, kind of a poorly worded question. So this is talking about the structure that has to form in order to actually exchange genetic material. A is correct, chiasma. Right? You have to do synapsis. But to actually, so synapsis comes first, right? Synapsis is gluing these chromosomes together. But to actually exchange genetic material, the chiasma or the chiasma actually has to form, which is the physical connection, which allows for those genetic combinations to be exchanged. So that's why 6A is the correct answer. Question 7. What is the synaptonemal complex? The correct answer is A. Oops. So it's the protein structure that forms between homologous chromosomes. B is not correct because it's saying sister chromatids, and nothing happens between sister chromatids. All the exchanging of material is going to be through your homologous chromosomes. So that's why B is wrong. Actin filaments, that doesn't have much to do with the synaptol complex. And then cleavage furrow, that's going to deal with cytokinesis. So again, not the synaptonemal complex. Okay, question 8. Homologous chromosomes are 100% identical. True or false? The correct answer is false, right? Because you get one from your mom and one from your dad, and they're different. All right, question 9. Sister chromatids are 100% identical. This one's a bit misleading. So we'll say for this one we're talking about mitosis. Sister chromatids are 100% identical in mitosis. True or false? Correct answer is true. And why I say this is a bit misleading is because if we're talking about meiosis, meiosis too, we're dealing with sister chromatids, and these will be unique because they just undergoed crossing over, and that would be not identical. So that creates variation. But if we're talking about mitosis, the sister chromatids are 100% identical. If we're talking about meiosis, then it's a different story. They're not 100% identical. Okay, number 10. If a turtle has 80 chromosomes, how many chromosomes are in its sperm? So let's just walk through this question, because this one can be a little challenging sometimes. So we're saying the turtle has 80 chromosomes. How many chromosomes are in its sperm? So the turtle itself has 80, so we're going to say diploid equals 80. Remember, diploid we abbreviate to 2N. Now, sperm, this could be sperm or egg. Sperm or egg are going to be our germ cells. Germ cells are always haploid. So haploid again is 1N, right? So if it's diploid 80, and we're talking about sperm cells, how do we get, what is our haploid of 80? Haploid again is N, so it should be pretty obvious, it should be 40. So the correct answer is in fact C. I can totally see this type of question being highly testable. This is just trying to give you an application question of, do you know what the difference between your diploid and your haploid cells are? Your somatic versus your germ cells. That's what this question is an application of. That is it for worksheet number 2. If you have any questions, let me know. Keep working hard, guys. Bye.